We're going to move now to some more functional talks. So first one is iPSC models of SYNGAP1 dysfunction from MARCELO COBA at USC. Thanks, Marcelo.
Thanks to the organizers. Really appreciate it. It's nice to be here and talk with many of you that I know for many years in person. I'm going to mainly discuss some topics on the use of SYNGAP on IPS models of disease. Yep. So, okay, you've seen a picture like this already. They just depicting SYNGAP, the cartoonish view of SYNGAP with many of these protein domains, modules of functions, and I'm going to discuss mostly what are we doing with focus on in RAS-GAP domain, which is this yellow domain in the middle, and the PDZ ligand function of SYNGAP, which is in purple and red circle to the right, and not many what are all other domains doing. But first, I'm going to just discuss this issue of the RAS-GAP function, RAS-GAP, RAG-GAP, RO-GAP, which all of them is being modulated by SYNGAP. And this idea that you can see here in this cartoon that at the end of the day, when you have a mutation in SYNGAP, you end up having an increase in phosphorylation of ERK2 of MAP kinase 1 that you've seen from many years, from Western blotted in some of the papers. So RAS-GAP tends to produce more GDP instead of GTP. In this case, it's RAS GDP or RAS GTP. It can be RAG GDP or RO GDP. At the end of the day, you get a signal cascade that ends up in this green name at the bottom. This is MAP kinase 1, which is ERK2, which is it measures protein phosphorylation. So the idea is that you have less SYNGAP, you have an increase in ERK2 phosphorylation. So we know that SYNGAP is mostly localized for many years, and the enrichment of SYNGAP is in mature synapses. In this EM picture, which is in gray there, you see this electrodense region, which is a darker gray, and synaptic vesicles. Sorry, but I cannot point, but you have these circles at the top. This is the presynaptic site. Opposite, you have the postsynaptic site, and in this electrodense region, you have SYNGAP there. So in theory, this RAS-GAP function is mostly functioning there, right? So all the experiments that have been done and shown this increase in ERK phosphorylation of SYNGAP haven't been done there, are not from there. We don't have any idea if SYNGAP, the RAS-GAP function, what is it doing, or if it works at the synapse. What it has been done is just get a lyset of neurons, and you do a Western blot, and you see increase in ERK phosphorylation. What it means is you have a dysfunction of SYNGAP, and at the end of the day, this neuron respond increase in ERK phosphorylation. Where, we don't know, and we think not mostly at the synapse. So it's not really proven a function of the RAS-GAP domain at the synapse. In fact, there's going to be a paper soon showing that a knockout model for the RAS-GAP domain doesn't do mostly anything at the synapse. Okay. Next function that we are going to at least get some ideas and show some data, the PDZ. The PDZ function is just on one of the isoforms. As you know, it's been called alpha-1 for many years. I'm not going to change the name. And it binds to these green modules, which is PDZ. So there are proteins that have these modules that are called PDZ domains. So SYNGAP has the property, at least one of the isoforms of SYNGAP has the property to bind to these protein modules. And there's a very well-known family of proteins that is called MAGOOC there, like you can see DLG1, DLG2, DLG3, and DLG4, which you will know at PhD 95. There are other family members, and I don't know if Marcelo is going to show something on the next talk, but we know that binds to other family members and outside the synapse. So we know a lot about this, what is it doing, and we know that in mature rodent synapses, it has a very well-described function. So if you mutate that module, the synapses, what will I say, you have a really, most of the phenotypes that you observe with the haplosufficient model of disease. So that's a pretty important function that this has been demonstrated. So this is just to show what it means, this PD set. So again, this is a problem, I cannot point to this, but if you look at this, the second row of all these letters, which show the C-terminus of different isoforms of SYNGAP1, the second is the alpha-1. And if you see these Q, which are glutamines in blue, that's the last, is the point of splicing from the last exon, so that's the difference between these isoforms. The only one that we know what is it doing is really the alpha-1. For all the others, we don't know what are they doing. They cannot bind for sure to PD set containing proteins, so if whatever they are doing is mostly related to the RAS-GAP function. But that's an assumption. We don't know, and it's pretty difficult to know what these other isoforms are doing without changing the total protein levels, because you don't need to mutate each of the isoforms. What you really need to do is to change this C-terminal domain to analyze really what are they doing. And it's been tricky. At least nobody did it. So we don't really know that. But that's generally, that's complicated. And SYNGAP is one of at least in mature rodent synapses. Also in humans, we have data in humans about what proteins are binding there. It's one of these more than 2,000 proteins at the postsynaptic site, and it's binding just what we showed, that it's binding to these PD set containing proteins. These are pretty important modules to get these protein-protein interaction networks at the synapse, what we've been studying for many years. So SYNGAP is in this synapses is one of the most abundant proteins, and the stoichiometry, it's important in terms of the proteins to what SYNGAP binds also at the PhD in mature rodent synapses are also pretty abundant. So what do we do now? We are still working with mouse models. We're going to show some data what we found in human models. First things, well, human models and the synapse and the types of neurons that we are going to generate. These are, let me see what it comes next. Okay. These are very different neurons that we've been studying SYNGAP for many years. These are fetal-like neurons. These are not the same as the rodent synapses that we are studying. Every data that we are going to collect here, it resembles a fetal neuron. We are doing, and this is important because the protein composition of these neurons and of these synapses that we quantitated is not the same as the one that we can find in a mature rodent synapses. So we are assuming that all the protein interactions are the same. We are assuming that the functions are the same, but we don't know. So we are doing, in general, at the lab, three types of cells. In this case, we do excitatory neurons, inhibitory neurons, and we are doing astrocytes. And we're going to show data just evaluating three functions of SYNGAP, what it happens with you have mutations that truncates the protein, and you have a decrease in the total protein level, or when you have changes in the RAS-GAP activity. Not as this, well, can be assumed as a BUS, but well, we are going to show just one particular example, and mutations that affect the PD set ligand. So in general, we like to evaluate what is it do when if you change the protein or you change the most studied functions of SYNGAP. And this is what I said. I said before. So you may ask why we are studying this now if in the past we were always studying in the mature rodent synapses. And in general, there are two main things that we like to see, mostly to study the genetic background of the patient, and also to study mechanisms of development, and for sure with any tool that might come in the future to trying to increase total protein levels, well, we want to study within the context of the human genetic background, and also you need the genetic elements of the human genome. So in general, what is happening when you have these types of mutations? So these are those nice pictures to show, and the next one is going to be more clear. But what happens when you have a truncation of SYNGAP and you have a reduction of the total protein levels, you have an accelerated differentiation, accelerated maturation of the neurons. So this is the case for excitatory neurons, and what you see at the top panel is measuring spine density of spine volume. Spine density, what it means is like, well, you are counting the number of spines. Spine volume, how big are these spines? So you have more spines and bigger spines when you have less total protein levels of SYNGAP. the bottom panel is showing what happens. Let me see. Yes, what it happens with a mutation in the RAS gap domain. So this is a point mutation in the RAS gap domain, so the RAS gap domain doesn't function, and we have the same phenotype. You have an increase in the maturation with more spines and larger spines. So in this developmental model, the RAS gap function is important. And it gets you a similar phenotype as having less total protein of levels. This is just to have a picture, because showing a picture is much better, really, with showing the quantitative model, the quantitative pictures, and the p-values. So at the top panel, you have a control. So it's an isogenic line from a patient that has a mutation, a truncation mutation in SYNGAP, and you can see the figure to the left. These are inhibitory neurons. These are GABAergic neurons, not excitatory neurons. That the cells are small. Well, the processes are smaller. So if you have, you see the lengths of the processes are smaller, and you cannot see the number of spines. Well, I can see them from here, but you have more spines, and they are bigger. You got to believe me, but at least you can see that the processes are bigger. So if you see those is much bigger, and this is a striking difference. You don't even need to do this. These are neurons with green fluorescent proteins, so you can look just at the microscope and you just see the difference. At the bottom panel, you see the PDZ mutation. So we mutated just 2 amino acids at the PDZ, at the C termini of SYNGAP. With this mutation, this is the same that has been done in the mouse model. So this PDZ cannot bind, this SYNGAP cannot bind to PDZ proteins. And with just that, it has the same phenotype as having a truncation in the protein or not having. So the PDZ also is pretty important. So in these models, we've seen the same phenotypes with the PDZ, mutations in the PDZ, with the RAS gap domain or not having protein. So and there's not much time to discuss things, but also this is just to show that, yes, this any acceleration in the differentiation of these GABAergic neurons. If you see at the bottom, you have different time points, 7 days, 14 days, and 21 days in vitro. And what is showing you in green is a control, and in red is a mutation in the PDZ ligand region of SYNGAP1. And if you see in the red dots that you have more at each time point to reach a plateau at 21 days, these are GABAergic neurons. So they are appearing, you are getting GABAergic neurons faster when you don't have the PDZ ligand than the control. So it's another way to show the differentiation. I don't have much time. This is electrophysiology recordings. There's not much relevant with electrophysiology recordings. Again, it's showing you this acceleration in phenotype. What you have is an increase in activity. You have an increase in activity because you have more spines and the spines are bigger. So if you want just to quantitate as an endpoint and you have any method that you are increasing total protein levels or changing the SYNGAP levels, it's up to you what are you going to use as a screening method, but electrophysiology is much more complicated. I would say it's better to go with the quantitation by microscopy. But also it's more complicated if you see also for the interpretation at the bottom panel is a co-culture of inhibitory and excitatory neurons. So the inhibitory neurons are going to inhibit the excitatory neurons, and again it's showing the same. When you have these mutations in SYNGAP, the inhibitory neurons inhibit more, so you have less activity. But this is all related to this increased differentiation. This is just biochemistry, just to show that, okay, what these synapses are bigger, what do they contain? So we isolated the postsynaptic compartment of these neurons, inhibitory or excitatory neurons, with the mass spectrometry. And yes, they contain more of these synaptic proteins there as well, more receptors, more scaffold proteins, so they are more mature. And just to end, we showed that changes in the amount of protein have a phenotype that is increasing the development, in differentiation, RAS gap domain mutations as well, and PDZ ligand mutation, but it's not always the case. We have patients that they don't observe this phenotype. Even if you have a truncation and you have less protein, the phenotype can be reversed. In this case, we have mutations that increases decreases the synaptic activity. And also, when you check on the morphology of the neurons, is different. You can see that this is genetic background. What is it in the genetic background? You know, it might be the total protein levels. When I say protein levels, we do mass spectrometry to get absolute quantitation of each isoform. We know that, for example, this cell has the amount of protein of Syngap total protein is different. We said it has a bit more, if that's related or not. But just to point that it's not always the same case. It's not you shouldn't expect the same. And we have the reverse background for each one of them, and we are studying the genetic background for this. But I think it's important just to remember that we don't expect to have the same. And when we are saying that most of these models are haplosufficient, we should measure if they're really what it means haplosufficient. The model that I show you is haplosufficient with the mass spectrometry, and they have 50% of the total protein levels. This is just funding, and what I really want to thank is that the Syngap family. I know many of you for many years. I have iPS cells from many of your kids. You have been a tremendous help, not just for the lab, but also moving ahead the field. Thank you.